Hello, and thank you for joining us for this webinar entitled Peptide Design Strategy, Basics, Optimization, and Application. I'm Tiffany Campolongo, and I'm a Regional Manager for Service Marketing here at GenScript. A few months ago, I gave a webinar called Avoiding Peptide Assay Failure, Hidden Problems and Solutions, where I discussed some general intrinsic properties of peptides that can cause your assays to fail. In this webinar, I'll expand on that topic, but in a more detailed fashion, as I'll explore general peptide design considerations, as well as highlight some of the latest trends in peptide design for a few research applications. This webinar will give you a better idea about what we believe are the most important parameters to consider during basic peptide design. Most importantly, I hope it will inspire you to try new strategies as you design novel peptides. If you have any questions during this presentation, you can submit them by typing them in the questions field currently showing on your screen. At the end of the presentation, I'll do my best to answer some of the questions. Frequently asked questions from the webinar will be posted with answers on our website at www.genscript.com/webinars. In addition, this webinar video and a PDF version of the slides will be archived on our website in about a week's time. Also, immediately after this webinar is over, a survey will appear on your screen. If you have a minute or two, please fill it out. Your feedback really is an invaluable resource that we use to improve our webinar series as well as the services we provide to you. So this presentation will have four major sections. I'll start with a brief discussion on why we design custom peptides. Then I'll discuss what we consider are the peptide design basics. These are general considerations and precautions that should be taken when designing any custom peptide. Then I'll discuss what we're calling advanced design strategies. This section will contain a little heavier subject material that is supported by case studies from peer-reviewed literature on trends in peptide design for specific research application areas. Throughout the presentation, I'll be highlighting services and tools that GenScript offers that can help you with your peptide design. And I'll end the presentation with a summary of the services and resources that GenScript has to offer, followed by the question and answer period. So I'd like to start out by talking about why researchers design custom peptides. This is usually governed by the final application of the peptide, and there are numerous diverse applications. In structural biology, a peptide is usually designed to represent the functional part of a protein. So it's designed to mimic a portion of the native protein sequence and may be used to assess catalytic activity, binding sites, or structural stabilization components. In drug discovery or development, peptides may be designed as protein or enzyme inhibitors or receptor agonists to alter cell signaling pathways. For instance, GPCRs are still a major peptide drug target. Or peptides may be designed to inhibit bacterial proteins or enzymes that result in the death of microbes. In vaccine development, peptides are usually designed to mimic the immunogenic portion of a viral protein so that it will elicit a particular immune response to HIV, HPV, or influenza. And in drug delivery, peptides may be designed to penetrate cells and may have elements that allow them to encapsulate or be conjugated to DNA or a small molecule drug, and then later release their cargo. Peptides are also designed to generate custom antibodies. If you want to learn in specific detail about how to design peptides for this purpose, we have an archived webinar on our website that you can download called Protein or Peptide Antigen, Choosing the Optimal Immunogen for Antibody Production. Finally, peptides are designed to self-assemble into structures that resemble the extracellular matrix and support cell or tissue growth in the design of biomaterials. Now keep in mind that this list isn't exhaustive by any means, as peptides have applications in many other research applications, such as proteomics and nanomaterials. Due to the myriad of applications for custom peptides, it's very difficult to come up with a one-size-fits-all strategy. But we believe there are 3 basic parameters that you should take into consideration when designing your custom peptide. The first is basic biochemistry. A peptide is a biopolymer that should be designed with the basic rules of amino acid biochemistry in mind. The second, and likely the most overlooked, is the chemical process of peptide synthesis. The most thoughtfully designed peptide won't be effective if it can't actually be synthesized at the quantity and purity you need for your experiments. So the peptide synthesis process must be kept in mind. If you need a brief refresher on the peptide synthesis process, you can download my previous webinar, Avoiding Peptide Assay Failure. Finally, application is most important. Considering what role your peptide will play in your experiment and what environment it will be assayed in plays the biggest role in your design strategy. Now, during your registration for this webinar, you had to answer a survey question, and that question asked, what is your biggest concern when designing custom peptides? And the top 3 answers were, number 1, designing novel, effective sequences. Number 2, avoiding hydrophobicity. And number 3, designing peptides that can be delivered on time. So the first section of this presentation on basic design strategy will address the last 2 concerns, avoiding hydrophobicity and designing peptides that can be delivered on time. The second section will review trends in novel sequence design. So let's get into the first topic, peptide design basics. As I mentioned before, a peptide is a biopolymer, and thus charge is a very important parameter. Charge can affect how a peptide will function in your assays. It can also govern the solubility of your peptides. In addition, some charged residues have a high affinity for contaminants in the peptide synthesis process, like TFA and water molecules. Charge is dependent on the ionizable groups within your peptide. Those groups include the terminal groups, so the N-terminal amine and the C-terminal carboxyl group, as well as some of the R groups of the amino acid sequence. Aspartic acid, glutamic acid, histidine, cysteine, tyrosine, lysine, and arginine are all amino acids that have ionizable R groups, meaning they can gain or lose electrons. Whether or not an ionizable group gains or loses an electron is based on the pH of its surrounding environment. Every peptide has a calculable isoelectric point, called its pI for short. The pI of a peptide describes the pH at which the overall charge of the peptide is neutral. Now there's an important relationship between the pI of a peptide and the pH of its surrounding environment. When the pH is greater than the pI, the peptide has a net negative charge. When the pH is less than the pI, the peptide has a net positive charge. But when the pI equals the pH, the peptide is neutral and has minimal solubility. It will be difficult to dissolve and may precipitate out of solution. For this reason, it's important to consider the buffer or solvent that your custom peptide will be assayed in, as well as calculate its net charge as you're designing your peptide. I'll talk about how you can calculate net charge in the next slide. As I mentioned, charge can also affect peptide activity. This is especially true if you're designing your peptide to mimic a portion of a protein. Remember, the amine and carboxyl groups of your peptide are ionizable. In particular, you should make sure that the N and C terminal ends of your peptide properly represent the potential ionization of the N and C terminal ends of the protein you're studying. If your peptide is derived from the N-terminal end of a protein sequence, you'll want to cap the C-terminal end of your peptide so that it mimics what would be an internal amino acid of the protein. Similarly, if your peptide is derived from the C-terminal end of a protein sequence, you'll want to cap the N-terminal end of your peptide. And if your peptide is derived from an internal stretch of protein sequence, you'll want to cap both the N and C terminal ends of your peptide, since neither would be exposed in the native protein. Capping is largely performed by acetylation of the amine group and amidation of the carboxyl group, which prevents ionization of the capped group. So along with considering the net charge of your peptide for solubility purposes, you should consider whether or not you should cap the N and or C termini of your peptide. And we'll revisit capping in the next slide. Now, there's one more charge-based relationship for you to consider. Certain charged residues have a high affinity for contaminants that are derived from the chemistry of the peptide synthesis process during production. For instance, lysine, histidine, and arginine bind trifluoroacetic acid, or TFA, which is a strong acid used to cleave a peptide from its solid resin. Our production team has found that TFA can comprise up to 45% of a lyophilized peptide preparation, and TFA has been found to affect cell viability, tissue viability, and enzyme activity. In addition, lysine and arginine bind water molecules, which can result in cleavage or degradation of your peptide. And tyrosine and glutamic acid bind protective groups, which can change the color of your peptide. So in summary, these contaminants can negatively affect the activity of your peptides or your peptide stability. For a more detailed discussion on each of these contaminants, again, you can reference my previous webinar on avoiding peptide assay failure. The steps for evaluating charge and its effect on your custom peptide consist of first calculating either the isoelectric point or net charge of your peptide. You can do this by hand, or you can do it using GenScript's peptide property calculator. To calculate net charge by hand, you would assign a value of -1 to acidic residues and the C-terminal carboxyl group. You would assign a value of +1 to basic residues and the amino terminal of the peptide, and then you would add all the charges together to calculate the net charge. GenScript has a handy do-it-yourself solubility chart, which you can find on our website, to determine what solvent would be the best to dissolve your peptide. If the charge is positive, the peptide is basic and should be dissolved in an acid like acetic acid. If the charge is negative, your peptide is acidic and should be dissolved in a base like ammonium hydroxide. And if the charge is 0, the peptide is neutral and should be dissolved in an organic solvent like acetonitrile, methanol, or isopropanol. Alternatively, GenScript's peptide property calculator will calculate the pI of your peptide and also give you recommendations on how to best dissolve your peptide. Secondly, you should decide on a capping strategy, selecting acetylation, amidation, or both, so that the potential for ionization of the N and C terminal groups is consistent with the stretch of protein represented by your peptide. Thirdly, if you're considered that the charge of your peptide will complicate your experiments, you may choose to alter your peptide sequence by making changes only to non-essential residues. Along with charge, peptide hydrophobicity can obviously influence solubility. Since hydrophobicity is one of the top concerns in peptide design as determined from the registration survey, I'm going to briefly go through some rules and tips here. Hydrophobic peptides are typically designed to represent membrane proteins, but they can also be used in a number of other applications. For example, they may be used as building blocks for self-assembled biomaterials. The problem with hydrophobic sequences is that it's more energetically favorable for them to interact with themselves than water, and so they minimize their interactions with water. For this reason, they are difficult to dissolve and may easily precipitate out of solution. The rule of thumb is that typically peptides will be hydrophobic if they're greater than 5 amino acids long, and over half the sequence contains hydrophobic residues like tryptophan, isoleucine, phenylalanine, methionine, valine, and tyrosine. Hydrophobic peptides do tend to take longer to synthesize and deliver since they can be difficult to purify. So if you find that your peptide fits this rule of thumb, you can decrease hydrophobicity by substituting non-essential amino acids with polar or charged amino acids. If you can't change your sequence, but can add to your sequence, you can add a string of charged amino acids to the end of your peptide, like a string of lysines, to help increase solubility. Designing a less hydrophobic peptide may decrease the turnaround time for your peptide, as well as save you the trouble of dealing with insoluble peptides. Now, if you can't reduce the hydrophobicity of your peptide by design, and you struggle with peptide solubility, I urge you to request GenScript's free solubility testing. Dissolving hydrophobic peptides can be a pain. It's time-consuming, and you could lose a lot of your custom peptide, several milligrams in fact. Our solubility testing service is for purified peptides and can be requested through our online quotation system or by email. After synthesizing your peptides, our scientists will attempt to systematically dissolve your peptide in a minimum of 3 solvent types: an aqueous solution, a buffer, and an organic solvent. We have a list of solvents for each of the 3 solvent types, and if your peptide doesn't dissolve in the first solvent, we'll move on to the next until we find a solvent that it will dissolve in. The final deliverable is a solubility test report, which will include the gross concentration of your peptide in at least 3 solvents. Solubility testing can save you time, money, and most importantly, it will help you conserve your peptide stock. If TFA contamination is your concern, GenScript offers a guaranteed TFA removal service, meaning we can guarantee the amount of TFA left over in your peptide preparation. We use a proprietary counterion exchange method to switch the TFA salt in your peptide to either chloride, acetate, or formate ions. The result is that we can reduce the amount of TFA in your custom peptide to less than 0.1% for TFA exchange to HCl, or less than 0.5% for TFA exchange to acetate or formate. We highly recommend this service for peptides that will be used in cellular, tissue, or animal assays, or for peptides that will be used as APIs or in manufactured products. Again, if you'd like to get more information about TFA and its effect, you can view that previous webinar on peptide assay failure for a more detailed discussion on this topic. Another basic design consideration is length. Length is an important parameter to consider if you're concerned about designing peptides that can be delivered on time and at the quantity and purity you need. That's because as the length of a peptide increases, the purity of the peptide decreases. Peptides are synthesized through cycles of deprotection and coupling of the next amino acid to the growing peptide chain. With each cycle, the potential for unwanted side reactions or deletion sequences increases, which increases the amount of impurities in the crude peptide product. This makes purification of the target peptide from impurities difficult and decreases the overall yield of the purified peptide. In an ideal world, we could design all peptides to be no longer than 15 amino acids. But since that's not possible, synthesis techniques have been developed to synthesize longer peptides. GenScript has developed proprietary ligation technology, which allows us to join two shorter peptides to become one longer peptide. This allows us to produce long peptides greater than 100 amino acids in length. We also offer recombinant peptide synthesis, which harnesses the power of protein expression to produce peptides longer than 150 amino acids. Recombinant peptide synthesis is also ideal for peptides having more than 3 disulfide bonds. However, the requirement of special synthesis techniques will increase the turnaround time for your peptide, so it's really in your best interest to design peptides less than 50 amino acids if you desire a quick delivery of your peptide. Shown here is a case study that demonstrates the versatility and capability of GenScript's long peptide synthesis technologies. A customer requested synthesis of an active portion of human T-cell leukemia virus 1 protease, which is a 126 amino acid long sequence that requires a complex secondary structure for activity. GenScript used a combination of solid phase peptide synthesis and ligation technology to produce the peptide. Afterwards, the activity of the peptide proved that it was an active protein capable of catalytic activity. The last parameter of basic design I'll discuss is peptide purity. The purity you select for your peptide is governed by the application. Here I've listed GenScript's recommendations for purity selection based on application. One thing that is important to note is that the hydrophobic and longer peptides are more difficult to purify. This will not only increase turnaround time, but may also decrease purification capabilities, which is an important consideration to keep in mind during peptide design. So now that we've discussed the basics of peptide design, we'll get into some more heavy lifting with what we're calling advanced peptide design strategy. Peptide stability is a key design consideration for peptides that are going to be used in either in vitro or in vivo assays. No matter how well you've designed your peptide to activate or inhibit its target, it will never be effective if it's not stable enough to make it there. Concerning peptide stability, you really need to design your peptide with two things in mind. One is that the peptide sequence you design could be subject to processes such as oxidation, hydrolysis, or deletions during the peptide synthesis process, or during storage of your peptide. The other part of your design strategy you should take into consideration is the application and environment that your peptide will ultimately be assayed in. In this case, you may need to focus your design on increasing cell permeability, avoiding protease degradation, or avoiding renal clearance. And I'll talk about some design trends for each of these categories in the upcoming slides. There are many sequence combinations that can cause your custom peptide to be unstable during the synthesis process. For instance, glutamine may undergo cyclization to pyroglutamate if it's located at the N-terminal end of your peptide during cleavage of the peptide from the resin. If your peptide has glutamate at the N-terminal end, it's recommended that you request acetylation of the N-terminal to prevent cyclization. Peptide oxidation is another common problem of peptides containing sulfhydryl groups, which is true of peptides containing cysteine and methionine. Oxidation of your peptide stored over time can decrease peptide activity, so it's important to store your peptides properly, and if necessary, substitute sulfhydryl-containing residues with non-sulfhydryl-containing residues of similar structure to reduce the effects of oxidation. In addition, certain sequence combinations such as glutamic acid, isoleucine, leucine, phenylalanine, threonine, tyrosine, and valine, or pairings of aspartic acid with glycine, proline, or serine can cause secondary structures to form or result in hydrolysis or truncated sequences. It's a good idea to avoid these pairings and combinations if possible to ensure the timely delivery of your peptide at the required quantity and purity. In vitro and in vivo peptides can be susceptible to proteases and/or peptidases. Exopeptidases such as aminopeptidases and carboxypeptidase target the N and C terminals of the peptide, while the internal sequence of a peptide is targeted by endoproteases like trypsin, chymotrypsin, pepsin, and elastase. Now, peptide susceptibility to proteases has been shown to correlate with its N-terminal residue. Peptides having methionine, serine, alanine, threonine, valine, or glycine at the N-terminal end tend to have a longer half-life, while peptides having phenylalanine, leucine, aspartic acid, lysine, or arginine tend to have a shorter half-life. To predict how your peptide might be cleaved by proteases, you can use peptide cutter tools, like PepCutter, which is offered on the ß website, shown below. Strategies for imparting peptide stability against proteases include simple modifications like acetylation and amidation, as well as more complex chemistries like cyclization or hydrocarbon stapling. Another big trend in design of peptide mimetics is the design of peptide mimetics, such as peptoids and the incorporation of unnatural amino acids like D or beta amino acids. We'll review a few case studies featuring these techniques in the next slides. This study that was published in Nature Biotechnology earlier this year demonstrates the power of beta amino acid substitution in increasing peptide stability. The researchers modified the peptide backbone to be protease resistant by substituting beta amino acids for native alpha amino acids in the peptide sequence. Beta amino acids differ from alpha amino acids in that their amino group is attached to the beta carbon instead of the alpha carbon. To demonstrate this technique, the researchers altered a portion of the parathyroid hormone called PTH, which is a protein that binds to the parathyroid hormone receptor PTHR1. PTH is an agonist of PTHR1, which is a G-coupled protein receptor. Binding of PTH to PTHR1 regulates processes such as bone homeostasis. Thus, PTH is the main component of an osteoporosis drug. However, the drug requires pulsative administration due to the short life of PTH. To combat this, the researchers replaced every fourth amino acid in PTH with a beta residue. The addition of the beta residues altered the peptide backbone by incorporating CH2 units into the backbone, while retaining side chains of the native sequence. Though not a natural sequence, this arrangement still mimics an alpha helical structure, which is important because this structure is crucial for PTH-PTHR1 binding interactions. The researchers designed 6 different PTH analogs by shifting the position of the beta residues or by altering the total number of beta residue replacements in the peptide. When the PTH analogs were tested alongside the native PTH sequence in stable cell lines in rats, it was found that the EC50 values for those analogs were comparable to that of the EC50 values for the native sequence. One of the analogs was considerably more potent than PTH in terms of prolonging signaling from PTHR1. This was attributed to the longer half-life of the analog compared to the native sequence. The results suggested that the prolonged half-life of the PTH analog may prolong PTHR1 signaling, thereby reducing the need for pulsative administration of PTH-based drugs. Another strategy for imparting peptide stability against proteases is to incorporate D-amino acids into peptides. D-amino acids are stereoisomers, or mirror images of the L-amino acids normally found in proteins. They can impart resistance against proteases since their structures may not be readily recognized by proteases evolved to recognize L-amino acid structures. Incorporation of a single D-amino acid may be powerful enough to greatly enhance a peptide's stability, like in the case of vasopressin, an antidiuretic hormone used to treat diabetes. Vasopressin normally has a half-life of 10 to 35 minutes in humans, but in one study, when a single L-arginine was replaced with D-arginine, the half-life of the peptide increased to 3.7 hours. A growing trend is to design peptides that are completely composed of D-amino acids, which are sometimes called mirror image peptides. There are a number of examples to show that this technique can be successful, including the development of mirror image peptides for the treatment of a variety of diseases such as HIV and most recently Ebola. Mirror image peptides have also been developed to treat ailments such as heart disease. For instance, a mirror image antiarrhythmic drug called rotigapeptide was successful enough to enter clinical trials. If you're interested in incorporating unnatural amino acids into your peptide design, GenScript Peptide Modification Service offers over 300 peptide modifications, which include beta and D amino acids. And we also can perform other modifications that can bolster peptide stability, such as disulfide bridges and cyclization. Peptoids are another approach for imparting peptide stability. In peptoids, the R groups are attached to nitrogen atoms instead of the alpha carbon. This arrangement alters the peptide backbone, resulting in peptoids being minimally or not at all cleaved by major protease classes. In addition, peptoids are resistant to denaturation by chemical denaturants and temperature. One disadvantage of peptoids is that binding of the R groups to the nitrogen atoms reduces the number of hydrogens available for hydrogen bonding, which is necessary for some secondary structure. But residue selection can help overcome this constraint. At GenScript, we currently offer 20 peptoid residues as well as a number of modifications for our custom peptoid services. Another consideration that researchers design for is renal clearance by the kidneys. Peptides that are hydrophilic in nature, that are less than 25 kilodaltons, can be filtered by the kidneys. In addition, peptides are not easily reabsorbed through the renal tubule. The most popular strategies to decrease renal clearance include conjugation to macromolecules or polymers, with the most popular being polyethylene glycol, or PEG. Pegylation of peptides increases molecular size and can shield peptides from proteases in the immune system. And PEG has been deemed safe and effective enough to be FDA approved. PSA, HES, and BSA are other conjugates that have been shown to improve renal clearance. As I mentioned before, GenScript can offer over 300 modifications, and along with unnatural amino acids, we also offer polymers and macromolecule conjugates like PEG and BSA. Yet another trend in the design of stable peptides is the development of stapled peptides. Stapled peptides make use of hydrocarbon stapling. For hydrocarbon stapling, two unnatural amino acids are incorporated into a peptide of alpha helical structure. The unnatural amino acids serve as anchors for the hydrocarbon bond, which imparts the peptide with extreme conformational stability of its alpha helical secondary structure. In addition, stable peptides are cell permeable and have high specificity to their targets due to the maintenance of their alpha helical structure. So stable peptides are becoming very popular for the development of peptide drugs for targets that were once considered undruggable. In this study, published in PNAS this year, researchers developed a stapled peptide as an MDM2-MDMX inhibitor. MDM2 and MDMX are inhibitors of p53, which are overexpressed in cancer cells and result in halting apoptosis and cell cycle arrest. The peptide, called ATSP-7041, was tested in cancer cells and in human xenograft models. ATSP-7041 was found to be able to disrupt both MDM2 and MDMX interactions with p53, which resulted in the reactivation of p53 and the suppression of tumor growth. The final case study that I'll present here takes into account a number of important parameters, including stability, secondary structure, and peptide activity. This study was published in Nature Chemistry earlier this year, and in it the researchers take a novel approach to peptide design by developing a new kind of amino acid called a dithiol amino acid. And this amino acid kind of mimics cysteine, except that it has two sulfhydryl groups instead of one. Through disulfide bridge formation, these amino acids are able to form unique branched or cyclic secondary structures. The dithiol amino acids were tested by incorporation into inhibitor peptides designed for a serine protease involved in tumor growth and an acetylcholine receptor. The result was that the dithiol amino acids increased peptide activity by 7.6-fold for the acetylcholine receptor inhibitor and 40-fold for the serine protease inhibitor when compared to the same peptide sequences containing natural cysteines. So, in summary, we covered a number of design considerations for custom peptides and presented basic and advanced strategies for each category. This was all based on current knowledge and what's trending now in the peptide research community. I hope that this discussion shed light on some design parameters that you had not considered before, as well as inspired you to explore new techniques as you design your next peptide. In addition, one key message I hope you take with you is that sequence evaluation is extremely important in peptide design, and it's worth evaluating your sequences and amending them if possible so that you can get stable, active, custom peptides delivered to you in a timely fashion. However, we know that in some cases your peptide sequence can't be amended, so GenScript has developed a number of specialized services that you can take advantage of and that should help make working with peptides easier. This slide presents all of the specialized services that we touched on in this presentation, which are all encompassed by our custom peptide synthesis service. Some of the services are free, such as solubility testing and acetylation and amidation capping, so I urge you to take advantage of those. And it's important to keep in mind that these services are not available post-peptide delivery. They have to be requested during the custom peptide quotation phase. So again, it's really important to evaluate your sequence beforehand so that you have an idea of what service you should request. Still, if you're unsure about whether or not you should request a specific service, we have a 24-hour technical account manager who are all former researchers, as well as our experienced peptide production team who are ready and willing to answer any questions you may have or help assist in your peptide design. In addition, I've highlighted some of the resources that GenScript offers to assist you in your custom peptide design. Don't forget to check out the 2 other webinars I mentioned throughout this webinar for even more tips on peptide design. So before I answer questions, I want to let you know that we'll have one more webinar in this fall 2014 series. That'll be on December 3rd at 8 o'clock a.m. and 2 o'clock p.m. Eastern Standard Time. And it's entitled Fusion Tag for Recombinant Soluble Protein Production in E. coli. In addition, I've listed the 2 additional webinars that I mentioned throughout this talk: Avoiding Peptide Assay Failure, Hidden Problems and Solutions, and Protein or Peptide Antigen, Choosing the Optimal Immunogen for Antibody Production. Both are available for download from our webinar webpage, so be sure to check those out if you haven't already. As I mentioned at the beginning of this presentation, the video of this webinar and a PDF version of the slides will be archived on our website in about a week's time. So please visit our webinar webpage to take advantage of these resources. And you can also email me directly at tiffany.campolongo@genscript.com with specific questions about this presentation. Also, please take a moment to complete a quick poll right after this webinar to tell us how you liked the presentation. Your candid feedback will be helpful for us in improving our services. So now I'm going to take a few questions from you guys. So the first question says, I got my peptide last week, but I'm having trouble dissolving it. Can I get a solubility test? So solubility testing is something that you have to request before you place your peptide order. We can accept peptides that are shipped to us for solubility testing. So what I recommend you do is visit the Peptide Solubility Guidelines webpage that's on our website and try to follow the guidelines there to try to dissolve your peptide. One option that you do have in the future is that you can request that we dissolve your peptide at GenScript before sending it to you, although this is not recommended because we can't guarantee the stability of your peptide in liquid form during shipping. But if you're really having trouble with solubility, you can request that. And be sure to request solubility testing the next time that you request a custom peptide. The next question says, is there a disadvantage to using D-amino acids? How about multiple ones? So this is a good question. Um, the one disadvantage I can think of to using D-amino acids, and this could probably be generally applied to any peptide um, with enhanced stability, is that the peptide may persist in the body too long and it could build up and cause toxicity. So there has to be a healthy balance between design for stability and the potential side effects that might accompany a really stable peptide. For multiple ones, it's not clear if there's a difference in advantage or different disadvantage to using single or multiple D-amino acids. The next question says, does GenScript provide norleucine unnatural amino acids? So the answer to that is yes. We have norleucine amino acid in stock. And if you'd like us to synthesize your peptide with norleucine, we can certainly do that for you. Okay, it looks like we have another question that says, what's the longest peptide you can produce? So the longest peptide that GenScript has ever produced has been 190 amino acids long. But as I mentioned before, long peptides are really difficult to synthesize because of that inverse relationship between length and purity. With that being said, GenScript routinely synthesizes peptides around 50 amino acids with ease. So if you have a peptide that's longer, our production scientists can certainly try to develop a synthesis protocol to help you synthesize that longer peptide. And we have a pretty versatile technology platform to do that. Um, so this is a housekeeping question. It says, can I get references for this presentation? And so when I create the PDF for this particular presentation, I will put in the reference list. And again, that version and the video version will be available from our website for download in about a week. So you can get the references at that time. Um, so we have one question that's asking about peptidomimetics based on a peptide sequence, but we don't have any chemistry. Can GenScript help us? I assume that you're asking if we can help you in the design of the peptide. So we don't necessarily have a service to help someone custom design the peptide, but if you have questions about peptoids or peptoid chemistry, certainly you could pose that question to one of our technical account managers, and they could check with our production team. They're all chemists there, and they could try to help you as best as possible to design a peptide according to the correct chemistry for peptoids. Okay, I'm going to take one more question, um, and that question is, will the slides be available after the presentation? And yes, again, as I said, the slides will be available for download after this presentation is over. It'll take about a week, and the archive video should be available for download as well. Okay, so that looks to be about all the time that we have. If I didn't answer your question, I will get the answer and post it on the website as soon as possible. And again, I thank you so much for joining me today. I hope you learned something, and I wish you all great success in your research.